Hello, and welcome to The Sleeper Scientist. I'm here to take you on a gentle journey through one of the most mysterious and beautiful phenomena in the universe, the story of how your brain writes, rewrites, and preserves the narrative of your life. Before we begin, I'd love to know where you're joining me from today and what time it is in your corner of the world. Also, don't forget to like the video and subscribe to the channel. Are you settling in for the evening? Perhaps you're in that peaceful twilight between day and night, where thoughts naturally turn inward and the mind begins to slow its relentless pace. I hope this exploration of memory isn't so fascinating that it keeps you awake. But if it does, at least you'll learn something remarkable about the very organ that's keeping you conscious. Imagine, if you will, that you're holding a book in your hands. Not just any book, but the most precious book that has ever existed. The complete story of your life. Every page contains moments both profound and mundane, emotions that have shaped you, faces that have touched your heart, and experiences that have molded you into who you are today. This book exists, dear listener, but not on paper or in digital form. It lives within the 3 pounds of neural tissue nestled safely within your skull. Tonight, we're going to explore how this most personal of libraries came to be, how it organises its vast collection, and why sometimes the most important chapters seem to be written in disappearing ink. We'll discover that memory isn't simply a recording device, like some biological video camera, but rather a skilled novelist, creative, selective, and occasionally prone to embellishment. As we embark on this journey together, I want you to let your mind drift, to allow your thoughts to flow like a gentle stream. Tonight, we're not in a hurry. We have all the time in the world to uncover the secrets of how your brain has been quietly, persistently, beautifully writing the story of you, the ancient understanding of memory. To truly appreciate the marvel of memory, we must first journey back through time, to understand how humanity has grappled with this invisible force that shapes our very identity. Close your eyes for a moment and picture ancient Greece, where philosophers in flowing robes gathered in marble courtyards, wrestling with questions that still puzzle us today. It was here, in this cradle of Western thought, that memory first began to be understood not merely as a practical necessity, but as something almost divine. The Greeks didn't just have one word for memory, they had an entire pantheon of concepts. There was Mnemosyne, the titaness of memory and mother of the 9 muses, who blessed humans with the ability to remember, and therefore to learn, to grow, to become more than mere animals driven by instinct. But the Greeks understood memory in ways that were far more sophisticated than we often give them credit for. They distinguished between different types of memory: mneme, the basic ability to retain past experiences, and anamnesis, the active process of recollection and remembrance. This distinction, made over 2,000 years ago, remarkably parallels modern neuroscientific understanding of different memory systems. Plato, in his dialogue Meno, explored one of the most profound questions about memory. How is it possible to learn something new? If you already know something, you don't need to learn it. But if you don't know it, how would you recognize it when you encountered it? His answer was anamnesis, the idea that learning is really a process of remembering truths that the soul already knows but has forgotten. While we no longer believe in Plato's theory of innate knowledge, his insight about the reconstructive nature of memory was remarkably prescient. When we remember something, we're not simply retrieving a stored copy, we're actively reconstructing the memory, and in doing so, we often discover connections and meanings that weren't apparent when the memory was first formed. What fascinated the ancient Greeks most was not just that we could remember, but how we remembered. They noticed something peculiar, something that would puzzle scholars for over 2 millennia. Memory wasn't like a wax tablet where impressions were simply pressed and preserved unchanged. No, memory was more like a living thing, growing and changing, sometimes becoming more vivid with time, sometimes fading like morning mist. Aristotle, that brilliant observer of human nature, was among the first to propose that memory work through association. He noticed that when he tried to recall something, his mind would often jump from one memory to another, connected by invisible threads. A smell might trigger the memory of his childhood home, which might lead him to remember his first teacher, which might then recall a particular lesson about mathematics. He called these chains of association, and though he couldn't have known it at the time, He was describing something that modern neuroscience would later confirm as one of the fundamental principles of how memory actually works. But Aristotle went further than simply describing how memory worked. He began to develop what we might consider the first scientific theory of memory. He proposed that memories were formed when sensory experiences created physical impressions in the brain, much like pressing a seal into warm wax. While the metaphor was imperfect, the underlying insight that memory must have a physical basis in the brain was revolutionary for its time. Aristotle also recognized that different people had different memory abilities, and he began to explore what factors might influence these differences. He noticed that younger people were generally better at forming new memories, while older people were better at retaining and accessing memories formed in their youth. He observed that emotional experiences were remembered more vividly than neutral ones, and that repeated exposure to information made it easier to remember. These observations, made through careful introspection and observation rather than controlled experimentation, were remarkably accurate. Modern neuroscience has confirmed that all of these factors, age, emotion, and repetition, do indeed influence memory formation and retrieval, and for reasons that Aristotle could never have imagined. The Roman contribution: memory as art. As Greek culture was absorbed into the Roman Empire, the understanding of memory evolved in new directions. While the Greeks had been primarily interested in memory as a philosophical problem, the Romans approached it as a practical art to be mastered and perfected. Roman rhetoricians like Cicero and Quintilian developed elaborate systems for memory training that were considered essential skills for any educated person. In a world without books, libraries, or any external storage systems, the ability to memorize and accurately recall vast amounts of information was not just impressive, it was necessary for survival and success. The Roman art of memory, or ars memoriae, was built around the principle of creating vivid, striking mental images and organizing them within familiar spatial frameworks. The most famous technique was the method of loci, or memory palace, which involved mentally placing items to be remembered in specific locations within a familiar building or route. But the Romans understood that effective memory was about more than just clever organizational tricks. They recognized that memory was enhanced by understanding, by emotion, and by practice. Quintilian advised that students should not simply memorize information mechanically, but should strive to understand its meaning and significance. He observed that memories formed through understanding were not only more accurate, but also more durable than those formed through rote repetition. The Romans also developed sophisticated understanding of the relationship between memory and rhetoric. They recognized that memory was not just about storing information, but about being able to access and deploy that information effectively in speech and argument. A good orator needed not just to remember the content of their speech, but to be able to adapt and modify that content in response to their audience and circumstances. This adaptive aspect of memory, the ability to modify and recombine stored information in creative ways, was something the Romans understood intuitively, but couldn't explain scientifically. We now know that this flexibility is one of the most remarkable features of human memory, made possible by the distributed and reconstructive nature of neural memory systems. The Roman approach to memory also emphasized the social and cultural dimensions of remembering. Memory was not just an individual cognitive ability, but a social practice that bound communities together through shared stories, traditions, and values. The great Roman historians like Livy and Tacitus saw themselves not just as recorders of facts, but as custodians of collective memory, responsible for preserving and transmitting the essential stories that defined Roman identity. Medieval monasteries, the cathedral of memory. As we drift forward through the centuries into the medieval period, we encounter a fascinating transformation in how memory was understood and cultivated. In the quiet scriptoriums of medieval monasteries, memory became not just a practical necessity, but a spiritual discipline, a way of bringing the soul closer to divine truth. Medieval scholars inherited the Roman techniques of memory training, but they adapted and expanded them in remarkable ways. The memory palaces they created were not just practical organizational tools, but elaborate spiritual edifices designed to house not only factual information, but also moral teachings, theological insights, and mystical experiences. Thomas Aquinas, perhaps the greatest medieval philosopher, wrote extensively about memory as one of the fundamental powers of the soul. He distinguished between memory proper, the ability to retain and recall past experiences, and reminiscence, the active process of searching for and retrieving forgotten memories. This distinction, like many medieval insights, proved remarkably prescient of modern neuroscientific discoveries about different memory systems. But what made medieval understanding of memory truly unique was its integration with spiritual practice. For medieval scholars, training the memory was not just about improving cognitive performance, it was about transforming the soul. They believed that by filling their memory with sacred texts, moral exemplars, and divine truths, they could gradually reshape their character and bring themselves closer to God. The memory techniques developed in medieval monasteries were extraordinarily sophisticated. Monks would create vast mental architectures, imaginary cathedrals, cities, and landscapes, populated with vivid characters and dramatic scenes that encoded theological and philosophical knowledge. These weren't just mnemonic devices, but elaborate works of mental art that took years to construct and perfect. They developed techniques for remembering not just what happened, but how it felt, not just the content of religious texts, but the emotional and spiritual states that those texts were meant to evoke. In doing so, they anticipated modern neuroscientific understanding of the intimate connection between memory, emotion, and meaning. Renaissance revelations and the birth of modern inquiry. As we move into the Renaissance, something profound began to shift in humanity's relationship with memory. The invention of printing transformed memory from a precious, scarce resource into something that could be externally stored and widely distributed. For the first time in human history, it became possible to preserve exact copies of texts and images, removing the burden of perfect memorization from individual minds. This technological revolution freed Renaissance thinkers to ask new questions about memory. Rather than focusing primarily on how to improve memory, they began to investigate what memory actually was, to probe the physical and psychological mechanisms underlying this most fundamental of mental faculties. Leonardo da Vinci, that supreme Renaissance polymath, approached memory with the same systematic curiosity he brought to anatomy, engineering, and art. His notebooks contain detailed observations about how memory worked, based on careful introspection and observation of others. He noticed that visual memories seemed to be particularly vivid and long-lasting, leading him to develop techniques for converting abstract information into visual form. Leonardo's anatomical studies also led him to speculate about the physical basis of memory. His drawings of the brain, while anatomically imperfect by modern standards, showed a sophisticated understanding that memory must somehow be encoded in the physical structure of the brain. He was among the first to reject the medieval notion that memories were stored in the fluid-filled ventricles of the brain, instead proposing that they must be encoded in the brain tissue itself. But it was the development of the scientific method during the Renaissance that truly began to transform our understanding of memory. Francis Bacon, one of the founders of modern scientific thinking, recognized that human memory was both a powerful tool for scientific investigation and a significant source of error and bias. Bacon observed that people tended to remember information that confirmed their existing beliefs, while forgetting information that contradicted them. He noticed that emotionally charged events were remembered differently than neutral ones, and that memories could be influenced by subsequent experiences and expectations. These observations led him to develop methodological principles for scientific investigation that were designed to minimize the distorting effects of human memory and bias. The Enlightenment. Memory as mechanism. The scientific revolution and Enlightenment brought yet another transformation in thinking about memory. As mechanical models of nature gained prominence, philosophers and early scientists began to conceptualize memory in increasingly mechanistic terms. René Descartes, the great French philosopher, proposed that memory worked like a mechanical system of traces and pathways in the brain. He suggested that when we experience something, it creates physical traces in the brain tissue, and that remembering involves reactivating these same physical pathways. While his specific proposals about brain function were wrong, his fundamental insight, that memory must involve changes in brain structure, was remarkably prescient. John Locke, the influential English philosopher, developed what may have been the first truly psychological theory of memory. Locke proposed that all knowledge comes from experience, and that memory is the mechanism by which past experiences influence present thinking and behavior. He distinguished between different types of memory: memory for facts versus memory for skills, recent memories versus distant ones, vivid memories versus faint ones. Locke's ideas about memory were revolutionary because they suggested that human nature itself was largely a product of memory. If all knowledge comes from experience, and if memory preserves and organizes that experience, then our personalities, beliefs, and capabilities are essentially the accumulated products of our memory systems. This insight would later prove crucial for understanding human development, learning, and psychological disorders. David Hume, the Scottish philosopher, pushed these ideas even further. Hume argued that our sense of personal identity, our feeling of being the same person over time, is entirely dependent on memory. Without memory to connect our past and present experiences, he suggested, there would be no coherent self, just a series of disconnected mental events, the birth of experimental psychology. The transition from philosophical speculation to scientific investigation of memory began in earnest in the 19th century. Hermann Ebbinghaus, whose work we touched on earlier, deserves a more detailed examination as the founder of experimental memory research. Ebbinghaus recognized that previous approaches to studying memory had been limited by their reliance on introspection and anecdotal observation. To truly understand memory, he believed, it was necessary to study it under controlled conditions using quantitative methods. But how could one study something as subjective and variable as human memory in a scientific way? Ebbinghaus's solution was ingenious in its simplicity. He created thousands of nonsense syllables, meaningless combinations of consonant-vowel-consonant, like dax or rup, that had no prior associations or emotional significance. By studying how he learned and forgot these artificial materials, he could isolate the basic mechanisms of memory from the complicating factors of meaning, emotion, and prior knowledge. Over a period of several years, Ebbinghaus conducted hundreds of carefully controlled experiments on himself. He would memorize lists of nonsense syllables, then test his ability to recall them after various delay intervals. He varied the length of the lists, the spacing of practice sessions, the amount of initial learning, and many other factors, meticulously recording his performance under each condition. The results of these experiments revealed several fundamental principles of human memory that remain valid today. Ebbinghaus discovered the forgetting curve, the mathematical relationship describing how memory decays over time. He found that forgetting is most rapid immediately after learning, then gradually slows down. He also discovered the spacing effect, the finding that distributed practice, spreading learning sessions over time, is more effective than massed practice, cramming everything into a single session. Perhaps most importantly, Ebbinghaus demonstrated that memory could be studied scientifically. His work established the foundation for experimental psychology and opened the door for the systematic investigation of mental processes that continues today. But Ebbinghaus's approach, while groundbreaking, also had significant limitations. By studying meaningless materials in artificial laboratory conditions, he was able to isolate basic memory mechanisms, but he may have missed some of the most important aspects of how memory works in real life. Real memories are embedded in rich networks of meaning, emotion, and personal significance, factors that Ebbinghaus deliberately excluded from his studies. This tension between experimental control and ecological validity continues to challenge memory researchers today. How can we study memory scientifically while still capturing its real-world complexity and richness? Early neuroscience, the brain revealed. While psychologists like Ebbinghaus were developing experimental methods for studying memory, physicians and anatomists were beginning to uncover the biological foundations of mental life. The 19th century saw remarkable advances in understanding brain anatomy and function, laying the groundwork for modern neuroscience. One of the most important early discoveries was the recognition that different brain regions have different functions. Paul Broca's studies of patients with brain damage revealed that specific language abilities were localized to particular brain areas. Carl Wernicke made similar discoveries about language comprehension. These findings suggested that complex mental abilities like memory might also be localized to specific brain regions. The first direct evidence for brain localization of memory came from studies of patients with brain injuries. In 1861, Théodule Ribot, a French psychologist, described what became known as Ribot's law, the observation that brain damage tends to affect recent memories more severely than remote ones. This suggested that memory was not uniformly distributed throughout the brain, but was organized in some systematic way. Even more intriguing were reports of patients who seemed to have very specific memory deficits. Some patients could remember faces but not names, others could remember events but not emotions, still others could learn new skills but couldn't remember learning them. These clinical observations suggested that memory might not be a single unified system, but rather a collection of different systems with different functions and different neural substrates. The development of increasingly sophisticated anatomical techniques revealed the extraordinary complexity of brain structure. The microscope revealed that the brain was composed of billions of individual cells, neurons, connected to each other in intricate networks. The Spanish anatomist Santiago Ramón y Cajal created beautiful drawings of individual neurons that revealed their elaborate branching structures and suggested how they might connect to form functional circuits. Ramón y Cajal proposed what became known as the neuron doctrine, the idea that the nervous system is composed of individual cells that communicate with each other at specialized junctions. This insight was crucial for understanding how memories might be stored in the brain. If memories were encoded in patterns of connections between neurons, then changes in these connections could provide a mechanism for learning and remembering. The emergence of psychoanalysis. As the 19th century drew to a close, a new approach to understanding memory emerged from an unexpected source: the treatment of psychological disorders. Sigmund Freud, working with patients suffering from hysteria and other psychological problems, began to develop theories about memory that would profoundly influence 20th century thought. Freud's insights about memory came primarily from his clinical observations of patients who seemed to have forgotten traumatic experiences. Through techniques like hypnosis and free association, Freud claimed to be able to help patients recover these forgotten memories, often with dramatic therapeutic results. Based on these clinical experiences, Freud developed a complex theory of memory that emphasized the role of unconscious processes and emotional conflicts. He proposed that memories were not simply passive records of past events, but active psychological forces that continued to influence behavior and experience long after the original events had occurred. Freud's concept of repression, the idea that painful memories could be actively excluded from consciousness while continuing to influence behavior, was particularly influential. This suggested that memory was not just about conscious recollection, but involved complex unconscious processes that operated according to different principles than conscious thought. While many of Freud's specific theories about memory have been challenged or discredited, his emphasis on the emotional and motivational aspects of memory was prescient. Modern neuroscience has confirmed that emotion plays a crucial role in memory formation, consolidation, and retrieval, and that unconscious memory processes do indeed influence behavior in ways that people are not consciously aware of. Freud also recognized that memory was not simply reproductive but reconstructive. He observed that when patients recovered supposedly forgotten memories, these memories often seemed to be influenced by their current psychological state and therapeutic relationship. This insight about the reconstructive nature of memory would later be confirmed and elaborated by experimental psychologists. Early 20th century, behaviorism and the black box. The early 20th century saw another major shift in approaches to studying memory as behaviorist psychology gained prominence. Behaviorists like John B. Watson and B.F. Skinner argued that psychology should focus on observable behavior rather than subjective mental processes like memory, thinking, and consciousness. From a behaviorist perspective, memory was not something that could be studied directly, since it involved unobservable mental processes. Instead, behaviorists focused on learning, the observable changes in behavior that result from experience. They developed sophisticated techniques for studying how organisms acquire new behaviors and how these behaviors are maintained over time. While behaviorists avoided talking about memory per se, their research revealed important principles about how experiences become encoded and influence future behavior. They discovered that the timing of rewards and punishments could dramatically influence learning, that different schedules of reinforcement produce different patterns of behavior, and that learned behaviors could be modified through various conditioning procedures. The behaviorist approach to studying learning and memory had both strengths and limitations. On the positive side, it brought scientific rigor and experimental control to the study of learning processes. Behaviorists developed precise methods for measuring learning, and discovered many fundamental principles that continue to inform our understanding of memory today. However, the behaviorist insistence on avoiding discussion of mental processes also limited their ability to understand the full complexity of memory. By treating the mind as a black box, whose internal workings could not be studied scientifically, behaviorists missed many important aspects of how memory actually works. This limitation became increasingly apparent as researchers began to study more complex forms of learning and memory. Simple conditioning models could explain some aspects of learning, but they seemed inadequate for understanding phenomena like language acquisition, problem solving, and the formation of complex memories for events and experiences. Medieval mysteries and Renaissance revelations. As we drift forward through history, we encounter the medieval scholars, those patient monks and Islamic philosophers who preserved and expanded upon ancient wisdom. In the quiet scriptoriums of medieval monasteries, scholars developed elaborate techniques for memory that seem almost magical to our modern minds. The memory palaces they created were architectural marvels of the mind. A scholar would imagine walking through a familiar building, perhaps the monastery where he lived, and in each room he would place the information he needed to remember. In the chapel, he might store theological arguments, in the kitchen, mathematical formulas, in the garden, poetry and literature. By taking a mental walk through these imaginary spaces, they could recall vast amounts of information with stunning accuracy. But here's where the story becomes truly fascinating, and this is something that wasn't understood until very recently. These medieval memory artists weren't just using a clever trick, they were unknowingly exploiting one of the most fundamental aspects of how our brains actually organize and retrieve memories. The hippocampus, that seahorse-shaped structure deep in our brain that we now know is crucial for memory formation, is also intimately connected to our ability to navigate space. When those medieval scholars created their memory palaces, they were essentially hijacking their brain's navigation system to create a filing system for information. What no one suspected at the time was that this connection between memory and space wasn't just a useful technique. It was a window into understanding how memory evolved in the first place. But that revelation would have to wait for the Renaissance and beyond. During the Renaissance, as anatomists began to peer inside the human skull, they discovered that the brain wasn't the simple, undifferentiated mass they had imagined. They found intricate structures, folded surfaces, and mysterious fluid-filled cavities. Some believed that memory lived in these cavities, that thoughts and experiences somehow floated in the cerebrospinal fluid like sediment in a pond. Leonardo da Vinci, that magnificent polymath, drew detailed anatomical sketches of the brain, trying to map the location of different mental faculties. He placed memory in the posterior ventricle, reasoning that it should be protected in the back of the brain, away from the sensory inputs that flooded in through the eyes, ears, and nose. His drawings were beautiful, his reasoning elegant, but like so many theories of his time, completely wrong. Yet in his curiosity, in his attempts to understand the physical basis of the mind, he was asking exactly the right questions. The dawn of scientific understanding. As we move into the 18th and 19th centuries, something remarkable began to happen. Scientists started to realize that memory wasn't just a philosophical curiosity, but something that could be studied, measured, and understood through careful observation and experimentation. Hermann Ebbinghaus, a German psychologist with an almost obsessive attention to detail, decided to study memory in the most rigorous way possible. He created thousands of nonsense syllables, meaningless combinations of letters like Vox and JIH, and spent years memorizing and forgetting them, carefully charting how his memory changed over time. What Ebbinghaus discovered would send shockwaves through the scientific community. Memory, he found, followed predictable patterns. Information was lost rapidly at first, then more slowly over time. He called this the forgetting curve, and it was the first mathematical description of how memory decays. But more importantly, Ebbinghaus had proven that memory could be studied scientifically. It wasn't just the domain of philosophers and poets anymore. It was a natural phenomenon that followed natural laws. But even as Ebbinghaus was mapping the landscape of forgetting, other researchers were beginning to suspect that the story was far more complex than anyone had imagined. Memory, they were starting to realize, wasn't just about storage and retrieval. It was about construction and reconstruction. and this discovery would eventually revolutionize our understanding of human consciousness itself. As the 19th century drew to a close, physicians began to encounter patients with strange and disturbing memory problems. Some could remember everything that happened before a certain date but couldn't form new memories. Others could learn new skills but couldn't remember learning them. These medical mysteries would prove to be the key to unlocking the deepest secrets of how memory actually works. But that part of our story must wait, for we've only just begun to understand the complexity of the memory system that evolution has gifted to us. The real revelations, the discoveries that would change everything we thought we knew about memory, were still to come. The architecture of remembering now, as we settle deeper into our exploration, I want you to imagine that you are an architect tasked with designing the most sophisticated library ever conceived. This library must be able to store not just books, but experiences, emotions, sensations, skills, and knowledge of every conceivable type. It must be able to retrieve any piece of information in milliseconds, must be able to make connections between seemingly unrelated items, and must be able to update and revise its contents constantly without losing essential information. Oh, and there's one more requirement. This entire library must fit into a space smaller than a cantaloupe and use less energy than a 20-watt light bulb. This impossible task is exactly what evolution has accomplished in creating the human memory system. And the solution it came up with is so elegant, so sophisticated, that we're only now beginning to appreciate its true complexity. Let's begin our journey into this neural library by understanding its basic architecture. Your brain contains approximately 86 billion neurons, nearly as many nerve cells as there are stars in the Milky Way galaxy. Each of these neurons can connect to thousands of others, creating a network of connections so vast and intricate that it defies comprehension. If we tried to map every connection in a single human brain, the resulting diagram would be more complex than a map of every road, path, and trail on Earth. But here's what's truly remarkable. This isn't a static library with fixed shelves and permanent locations for books. Instead, it's a dynamic, ever-changing system where memories are distributed across multiple brain regions, and where the very act of remembering changes the memory itself. Consider what happens when you try to remember your first day of school. The visual aspects of that memory, perhaps the colour of your classroom walls, the face of your teacher, the way sunlight streamed through the windows, these are stored and processed in your visual cortex, the same regions that process what you're seeing right now. The emotional content of that memory, whether you felt excited, nervous, or homesick, is processed in your limbic system, particularly in structures like the amygdala and hippocampus. The sounds you remember from that day, your teacher's voice, the chatter of other children, the squeak of chalk on a blackboard, these are processed in your auditory cortex. If you remember the smell of new textbooks or the feeling of unfamiliar school supplies in your hands, these sensory memories are processed in your olfactory and somatosensory cortices respectively. But here's where it gets fascinating. There's no single place in your brain where the complete memory of your first day of school is stored. Instead, when you remember that day, your brain recreates the experience by simultaneously activating all of these different regions and binding them together into a coherent recollection. It's as if your memory is like a symphony orchestra, with different sections playing their parts in perfect coordination to create a unified experience. The neuron building block of memory. But let's zoom in even closer to understand the basic building blocks of this remarkable system. Each neuron in your brain is a microscopic marvel of biological engineering. At its heart is the cell body, containing the nucleus and most of the cellular machinery needed to keep the neuron alive and functioning. Extending from the cell body are two types of projections. Dendrites, like the branches of a tree, receive signals from other neurons. The axon, often much longer than the dendrites, carries signals away from the cell body to other neurons. Some axons are microscopic, connecting nearby neurons. Others can be over a meter long, carrying signals from your brain down to your spinal cord and out to your muscles. The magic happens at the synapses, the tiny gaps between neurons where communication takes place. When an electrical signal reaches the end of an axon, it triggers the release of chemical messengers called neurotransmitters. These molecules drift across the synaptic gap and bind to receptors on the receiving neuron, potentially triggering a new electrical signal. But here's what makes this system so remarkable for memory. Synapses are not fixed, unchanging connections. They can be strengthened or weakened based on the patterns of activity they experience. When two neurons are repeatedly active at the same time, the synapse between them becomes stronger and more efficient. This is the physical basis of learning and memory, the encoding of experience in the strength of synaptic connections. Donald Hebb, the Canadian psychologist who first articulated this principle in 1949, put it elegantly: neurons that fire together, wire together. This Hebbian plasticity is the foundation of all learning and memory, from the simplest conditioned reflexes to the most complex human experiences. The dance of neurotransmitters. The chemical communication between neurons involves an intricate ballet of neurotransmitters, each with its own role in the memory process. Let me take you through some of the key players in this molecular drama. Acetylcholine, often called the neurotransmitter of attention and learning, is crucial for forming new memories. When you're paying attention to something important, neurons in your brainstem release acetylcholine throughout your brain, effectively telling your neural circuits that this information is worth encoding. People with Alzheimer's disease often have severely depleted acetylcholine systems, which helps explain their profound memory difficulties. Dopamine, famous for its role in reward and motivation, also plays a crucial part in memory formation. When you experience something unexpectedly pleasant or significant, dopamine neurons fire in bursts, releasing this neurotransmitter in brain regions involved in learning and memory. This dopamine signal acts like a biological save command, telling your brain that this experience is worth remembering. But dopamine's role in memory is more nuanced than simply marking important events. It also seems to be involved in updating and modifying existing memories. When you encounter something that violates your expectations, when reality doesn't match your predictions, dopamine neurons fire in complex patterns that can strengthen new memories while potentially weakening old ones that are no longer accurate. Norepinephrine, the brain's version of adrenaline, is released during times of stress, excitement, or emotional arousal. This neurotransmitter has a powerful effect on memory formation, which is why you tend to remember emotionally significant events much more vividly than mundane ones. The release of norepinephrine during emotional experiences activates the amygdala, which then influences the hippocampus and other memory systems to encode these experiences with enhanced strength and durability. GABA, gamma-aminobutyric acid, the brain's primary inhibitory neurotransmitter, might seem like an unlikely player in memory formation, but it's actually crucial for proper memory function. GABA helps to regulate the excitability of neural networks, preventing them from becoming overactive. In memory systems, GABAergic inhibition helps to sharpen the patterns of neural activity, allowing for more precise encoding and retrieval of information. Glutamate, the brain's primary excitatory neurotransmitter, is perhaps the most important neurotransmitter for memory formation. Most of the synapses in your brain use glutamate for communication, and changes in glutamate signaling are central to synaptic plasticity. The NMDA and AMPA receptors that respond to glutamate are the molecular machines that implement Hebbian learning, strengthening synapses when neurons are active together. The molecular machinery of memory. But let's dive even deeper, down to the molecular level, where the real magic of memory formation takes place. When you experience something worth remembering, let's say you're walking through a garden and you catch the scent of roses on a warm summer evening, something extraordinary happens in your brain. The neurons that are active during this experience begin to change at the most fundamental level. Proteins are manufactured, gene expression is altered, and new connections between neurons are literally grown. This isn't just a metaphor. Your brain is physically, structurally different after experiencing something memorable than it was before. The key player in this molecular drama is a protein called CREB, the cyclic adenosine monophosphate response element binding protein. When CREB is activated in a neuron, it triggers a cascade of genetic changes that can strengthen the connections between that neuron and its neighbours. Think of CREB as a molecular switch that determines whether a fleeting experience will be transformed into a lasting memory. But the story of CREB is more fascinating than you might imagine. Not all neurons express the same amount of CREB, and those with higher levels of CREB are more likely to be recruited into memory traces. This creates a kind of competition among neurons. Those with more CREB have an advantage in being selected to store memories, while those with less CREB are less likely to participate in memory formation. Recent research has shown that this competition can be manipulated experimentally. By artificially increasing creep levels in specific neurons, researchers can bias which neurons get recruited into memory traces. By decreasing creepy levels, they can prevent neurons from participating in memory formation. This suggests that the allocation of memories to specific neurons is not random, but is governed by molecular factors that can be influenced and controlled. But creep doesn't work alone. There's an entire cast of molecular characters involved in memory formation. There's PKM-zeta, protein kinase M-zeta, which helps maintain memories over long periods of time. This protein is particularly fascinating because it seems to be both necessary and sufficient for maintaining long-term memories. When PKM-zeta is blocked in brain slices, previously established memories disappear. When it's artificially activated, it can enhance memory storage. There's BDNF, brain-derived neurotrophic factor, which promotes the growth of new neural connections. BDNF is like fertilizer for synapses. It encourages them to grow stronger and more complex. Exercise increases BDNF levels in the brain, which may be one reason why physical activity seems to improve memory and learning. There's ARC protein, which travels from the cell body to the synapses like a molecular messenger, carrying instructions for synaptic changes. ARC is particularly interesting because it's one of the few neuronal proteins that is rapidly synthesized in response to experience and then travels to specific synapses where memories are being formed. Each of these proteins plays a crucial role in what neuroscientists call synaptic plasticity, the ability of connections between neurons to strengthen or weaken based on experience. This is the physical foundation of all learning and memory, and it's governed by principles that are both elegant and complex. Long-term potentiation, the cellular basis of learning. One of the most important discoveries in neuroscience has been the identification of long-term potentiation, LTP, a persistent strengthening of synapses based on recent patterns of activity. LTP was first discovered in the hippocampus in the 1970s, and it has since become the leading candidate for the cellular mechanism underlying learning and memory. LTP can be induced by high-frequency stimulation of synapses. When a synapse is activated repeatedly over a short period of time, it becomes stronger and more efficient. This strengthening can last for hours, days, or even longer, providing a cellular mechanism for the persistent storage of information. But LTP is not just about strengthening individual synapses. It involves complex changes in the molecular composition of synapses, the growth of new synaptic connections, and even the formation of entirely new dendritic spines, the small projections where most excitatory synapses are located. The induction of LTP requires the activation of NMDA receptors, a special type of glutamate receptor that acts as a coincidence detector. NMDA receptors only open when two conditions are met simultaneously. Glutamate must be present, indicating that the presynaptic neuron is active, and the postsynaptic neuron must be depolarized, indicating that it too is active. This requirement for coincident activity makes NMDA receptors perfect for implementing Hebbian learning. They only trigger synaptic strengthening when both neurons are active together. Once activated, MDA receptors allow calcium to enter the postsynaptic neuron. This calcium acts as a second messenger, triggering a cascade of molecular events that lead to synaptic strengthening. Calcium activates various protein kinases, which then phosphorylate AMPA receptors, another type of glutamate receptor, making them more effective. It also triggers changes in gene expression that lead to the synthesis of new proteins needed for long-term synaptic changes. But the story doesn't end there. LTP also involves structural changes to synapses. New AMPA receptors are inserted into the synaptic membrane, making the synapse more responsive to glutamate. The postsynaptic density, the protein-rich region where receptors and signaling molecules are concentrated, becomes larger and more complex. In some cases, entirely new synaptic connections are formed. These structural changes can be remarkably persistent. Some forms of LTP can last for weeks or months, providing a cellular mechanism for long-term memory storage. And because LTP can be induced repeatedly, allowing synapses to be strengthened in an incremental cumulative fashion, it provides a mechanism for the gradual acquisition of complex memories and skills. The hippocampus, master orchestrator of memory. At the heart of this complex memory system lies a structure that has captured the imagination of neuroscientists for decades, the hippocampus. Named for its resemblance to a seahorse, hippocampus is Greek for horse of the sea, This curved structure deep in the temporal lobe is perhaps the most intensively studied region of the human brain. The hippocampus serves as something like a memory conductor, orchestrating the formation of new memories and helping to bind together the various elements of an experience into a coherent whole. But it doesn't work like a simple recording device. Instead, it acts more like a sophisticated editor, selecting which experiences are worth preserving, how they should be organized, and how they relate to existing memories. The hippocampus has a unique anatomy that makes it particularly well suited for memory formation. It consists of several interconnected regions: the dentate gyrus, CA3, CA2, and CA1, each with distinct properties and functions. Information flows through these regions in a largely unidirectional manner, creating what neuroscientists call the trisynaptic circuit. The dentate gyrus is where most new information enters the hippocampus. It receives inputs from the entorhinal cortex, which itself receives information from throughout the brain. The dentate gyrus has a remarkable property. It continues to generate new neurons throughout life, a process called adult neurogenesis. These new neurons may play a special role in memory formation, helping to distinguish between similar experiences and reducing interference between overlapping memories. From the dentate gyrus, information flows to area CA3, which has a very different character. CA3 neurons have extensive connections with each other, creating what neuroscientists call a recurrent network. This recurrent connectivity allows CA3 to act as an associative memory system. It can store patterns of activity and later retrieve them from partial cues. The associative properties of CA3 are crucial for memory function. When you experience something new, the pattern of activity in CA3 becomes associated with that experience. Later, when you encounter a partial reminder of that experience, perhaps just a smell or a sound, the recurrent connections in CA3 can reactivate the full pattern, allowing you to retrieve the complete memory. From CA3, information flows to CA1, which acts as a comparator and output region. CA1 receives information both from CA3, representing retrieved memories, and directly from the entorhinal cortex, representing current sensory input. By comparing these two information streams, CA1 can detect mismatches between expectation and reality, signaling when something novel or unexpected is occurring. This comparison function of CA1 is crucial for learning. When there's a mismatch between what CA3 predicts based on past experience and what the entorhinal cortex reports about current sensory input, CA1 generates an error signal that can drive learning and memory formation. This is how the hippocampus helps you learn from experience, by detecting when your predictions are wrong and updating your memory accordingly. The temporal dynamics of memory formation. But the hippocampus doesn't just create memories, it also manages their transformation over time. When you first experience something new, the memory trace is heavily dependent on the hippocampus for its maintenance and retrieval. But over time, through a process called systems consolidation, memories gradually become independent of the hippocampus and are transferred to long-term storage in the neocortex. This transfer process can take years to complete, and during this time, memories remain malleable and subject to change. Each time you recall a memory during this consolidation period, it becomes temporarily unstable and must be reconsolidated, stored again. During reconsolidation, memories can be updated, modified, or even completely transformed based on new information or changing circumstances. The timeline of systems consolidation varies dramatically for different types of memories. Simple factual memories, like the capital of France or your phone number, may become hippocampus independent relatively quickly, perhaps within days or weeks. But complex episodic memories, like your memory of your wedding day or your first day of college, may remain partially dependent on the hippocampus for decades. This differential consolidation timeline has important implications for understanding memory disorders. Patients with hippocampal damage often show a temporal gradient in their memory loss. They can remember events from the distant past but not from the recent past. This pattern suggests that old memories have been successfully transferred to neocortical storage sites, while newer memories were still dependent on the hippocampus when it was damaged. But recent research has challenged the simple idea that memories are simply transferred from hippocampus to cortex during consolidation. Instead, it appears that the hippocampus may retain some role in memory storage throughout life, particularly for detailed episodic memories. The relationship between hippocampal and neocortical memory systems may be more of a partnership than a handoff. Pattern separation and pattern completion. The hippocampus also plays a crucial role in what neuroscientists call pattern separation and pattern completion, two complementary processes that are essential for proper memory function. Pattern separation allows us to distinguish between similar experiences and store them as separate memories. This is crucial because many of our experiences are quite similar to each other. You go to the same grocery store many times, you have dinner at home most nights, you drive the same route to work repeatedly. Without pattern separation, these similar experiences would interfere with each other, making it impossible to remember specific episodes. The dentate gyrus seems to be particularly important for pattern separation. Its sparse connectivity pattern, each dentate granule cell receives input from only a small percentage of entorhinal cortex neurons, helps to create distinct patterns of activity for similar inputs. The adult neurogenesis that occurs in the dentate gyrus may also contribute to pattern separation by providing new neurons that can help distinguish between similar experiences. Pattern completion, on the other hand, allows us to reconstruct complete memories from partial cues. When you hear a particular song, you might remember not just the music but the entire context in which you first heard it, where you were, who you were with, how you were feeling. This ability to retrieve rich, detailed memories from fragmentary cues is a hallmark of human memory. Area CA3 appears to be specialized for pattern completion. Its recurrent connectivity allows it to store and retrieve complete patterns of activity from partial inputs. When a subset of CA3 neurons is activated by a partial cue, the recurrent connections can reactivate the full pattern that was originally stored. The balance between pattern separation and pattern completion is crucial for memory function. Too much pattern separation would prevent you from generalizing across similar experiences or retrieving memories from partial cues. Too much pattern completion would cause interference between similar memories and make it difficult to remember specific episodes. The amygdala, the emotional modulator of memory. No discussion of memory systems would be complete without examining the amygdala, a small almond-shaped structure that plays a crucial role in emotional memory. The amygdala is best known for its role in fear and anxiety, but its influence on memory extends far beyond threatening situations. The amygdala acts as an emotional significance detector, evaluating incoming sensory information for its potential importance or threat value. When the amygdala determines that something is emotionally significant, whether positive or negative, it releases stress hormones and neurotransmitters that modulate memory formation in other brain regions. This emotional modulation of memory serves an important adaptive function. Events that trigger strong emotions are often events that are important for survival, threats to avoid, resources to seek, social relationships to maintain. By enhancing memory for emotionally significant events, the amygdala helps ensure that we'll remember and be able to respond appropriately when similar situations arise in the future. The amygdala influences memory through several mechanisms. It has direct connections to the hippocampus, allowing it to modulate memory formation at the source. It also influences memory through its connections to brainstem nuclei that release neuromodulators like norepinephrine, dopamine, and acetylcholine throughout the brain. When you experience something emotionally arousing, the amygdala triggers the release of stress hormones like epinephrine and cortisol. These hormones not only prepare your body for action, but also enhance memory consolidation. They activate the vagus nerve, which carries signals back to the brain, and they directly influence memory-forming regions like the hippocampus. But the amygdala's influence on memory is not uniformly positive. While moderate levels of emotional arousal enhance memory formation, extreme stress can actually impair memory. Very high levels of stress hormones can damage hippocampal neurons and disrupt normal memory processes. This is why people sometimes have fragmented or incomplete memories of traumatic events. The amygdala is also involved in a special type of memory called fear conditioning. When you experience something frightening in a particular context, the amygdala forms a strong association between that context and the fear response. Later, simply being in a similar context can trigger fear, even if there's no actual threat present. This fear conditioning can be remarkably persistent and difficult to extinguish. Once formed, fear memories seem to be stored permanently in the amygdala, although they can be suppressed by new learning. This persistence of fear memories may be adaptive in natural environments. where threats that were dangerous once are likely to be dangerous again. The prefrontal cortex, the executive controller. While the hippocampus and amygdala are crucial for memory formation and emotional modulation, the prefrontal cortex serves as the executive controller of memory systems. This region, located at the very front of the brain, is involved in the strategic aspects of memory, deciding what to remember, how to organize information, and when to retrieve specific memories. The prefrontal cortex is what allows you to engage in effortful strategic remembering. When you try to recall where you left your keys, when you study for an exam, or when you search your memory for the name of someone you met at a party, you're relying heavily on prefrontal cortex functions. Different regions of the prefrontal cortex seem to specialize in different aspects of memory control. The dorsolateral prefrontal cortex is involved in working memory, the ability to hold and manipulate information in consciousness for brief periods. The ventromedial prefrontal cortex is involved in memory retrieval and in evaluating the accuracy and relevance of retrieved memories. The prefrontal cortex also plays a crucial role in what psychologists call meta-memory, your knowledge about your own memory abilities and strategies. This includes knowing what you know and don't know, predicting how well you'll remember something, and selecting appropriate strategies for learning and remembering. Meta-memory is particularly important for academic learning and professional performance. Students who have good meta-memory are better able to allocate their study time effectively, focusing more effort on material they haven't yet mastered. They're also better at choosing appropriate learning strategies for different types of material. The prefrontal cortex undergoes prolonged development, not reaching full maturity until the mid-20s. This extended development period helps explain why children and adolescents often have difficulties with strategic memory tasks that require planning, organization, and cognitive control. The default network and spontaneous memory. Recent neuroscience research has revealed an intriguing aspect of brain function, the existence of a default network that is active when the brain is not focused on specific tasks. This network, which includes regions like the medial prefrontal cortex, posterior cingulate cortex, and angular gyrus, seems to be involved in spontaneous memory retrieval and mental time travel. When you're not actively focused on something specific, when you're daydreaming, letting your mind wander, or just relaxing, the default network becomes active. During these periods, memories often come to consciousness spontaneously, without any deliberate effort to retrieve them. You might suddenly remember a conversation from last week, a childhood experience, or something you need to do later. This spontaneous memory retrieval serves important functions. It allows you to consolidate and integrate memories, to make connections between different experiences, and to plan for the future based on past experiences. The default network seems to be constantly working in the background, organizing and reorganizing your memories and helping to maintain your sense of personal identity and continuity. The default network also seems to be involved in mental time travel, the ability to project yourself into the past or future. When you remember a past experience, you don't just recall facts about what happened, you mentally transport yourself back to that time and place, re-experiencing it from your past perspective. Similarly, when you imagine future scenarios, you mentally project yourself forward in time. This mental time travel ability is uniquely human and is crucial for many higher-order cognitive functions. It allows you to learn from past experience, to plan for the future, and to understand yourself as a continuous entity existing across time. Sleep and memory consolidation. One of the most fascinating discoveries in memory research has been the recognition of sleep's crucial role in memory consolidation. While you sleep, your brain doesn't just rest. It actively processes and organizes the day's experiences, determining which memories to strengthen and which to let fade away. During sleep, the brain goes through several distinct stages, each with different patterns of neural activity and different functions for memory processing. During slow-wave sleep, the deepest stage of non-REM sleep, the brain exhibits large, synchronized waves of activity that seem to coordinate the transfer of memories from hippocampus to neocortex. These slow waves are generated by the thalamus and spread throughout the cortex, creating periods of synchronized activity followed by periods of relative silence. During the active phases, the hippocampus replays the day's experiences, often at speeds much faster than they originally occurred. This replay seems to help strengthen important memories, while allowing unimportant ones to fade. REM sleep, characterized by rapid eye movements and vivid dreams, seems to play a different role in memory processing. During REM sleep, the brain shows high levels of activity in regions involved in emotion and memory, while areas involved in logical reasoning and reality monitoring are less active. This pattern may help the brain form creative associations between different memories and integrate emotional experiences. The timing of different sleep stages is crucial for optimal memory consolidation. Slow-wave sleep tends to predominate in the first half of the night, while REM sleep is more common in the second half. This timing seems to optimize the sequence of memory processing, first transferring and stabilizing important memories during slow-wave sleep, then integrating them with existing knowledge and forming new associations during REM sleep. Sleep deprivation has profound effects on memory function. Even a single night of sleep loss can impair the formation of new memories and reduce the brain's ability to consolidate memories from the previous day. Chronic sleep deprivation can lead to more severe memory problems and may contribute to age-related cognitive decline. The discovery of sleep's role in memory has important practical implications. It suggests that getting adequate sleep is not a luxury, but a necessity for optimal cognitive function. It also helps explain why sleeping on a problem sometimes leads to new insights. The brain's memory consolidation processes during sleep can help you see connections and solutions that weren't apparent when you were awake. The mystery of forgetting. Now, here's something that might surprise you. Forgetting isn't a flaw in the memory system, it's a feature. In fact, forgetting might be one of the most important functions your brain performs. Think about it for a moment. Every day you're bombarded with millions of pieces of sensory information. You see thousands of faces, hear countless sounds, smell numerous odors, feel various textures and temperatures. If your brain tried to preserve every single detail of every moment, you would quickly become overwhelmed by irrelevant information. Instead, your brain has evolved sophisticated mechanisms for filtering out the unimportant and preserving only what's likely to be useful in the future. This process of selective forgetting allows you to focus on what matters, while discarding the trivial details that would otherwise clutter your memory. Recent research has revealed that forgetting is an active process, not just passive decay. Your brain contains specific mechanisms designed to weaken and eliminate memories that are no longer needed. For example, there are enzymes that can break down the proteins that maintain synaptic connections, effectively erasing the physical traces of unwanted memories. This active forgetting system is particularly important during sleep. While you sleep, your brain doesn't just strengthen important memories, it also actively weakens and eliminates memories that are no longer useful. This process helps prevent interference between memories and ensures that your memory system doesn't become cluttered with irrelevant information. But forgetting serves another crucial function. It allows for generalization and abstraction. If you remembered every specific instance of seeing a dog with perfect clarity, you might have trouble recognizing the general concept of dog. Forgetting the specific details while preserving the general pattern allows you to build abstract concepts and categories that make learning and reasoning possible. The forgetting process is also crucial for emotional well-being. The ability to forget, or at least reduce the emotional impact of painful memories, allows you to move on from negative experiences and maintain psychological health. People who have difficulty forgetting emotional memories may be more prone to conditions like depression and post-traumatic stress disorder. However, the forgetting process is not random or uniform. Memories that are frequently recalled tend to be strengthened and preserved, while memories that are never accessed tend to fade. Memories that are emotionally significant are more resistant to forgetting than neutral memories, and memories that are integrated with other knowledge tend to be more durable than isolated facts. Understanding the mechanisms of forgetting has important implications for education and therapy. The digital age and memory. As we drift into the 21st century, our understanding of memory has entered a remarkable new phase. The same computational revolution that has given us smartphones and the internet has also provided neuroscientists with unprecedented tools for studying the brain. We can now watch individual neurons fire in real time, map the connections between brain regions with exquisite precision, and even manipulate specific memories in living animals. But perhaps more intriguingly, the digital age has begun to change memory itself. Think about how you use your smartphone. It's not just a communication device, it's an external memory system. You no longer need to memorize phone numbers, directions, or even important dates. Your phone remembers these things for you, freeing up your biological memory for other purposes. Psychologists call this cognitive offloading, and it's transforming how human memory functions. When information is readily available externally, we become less likely to store it internally. Instead, we remember where to find information rather than the information itself. Some researchers worry that this might be weakening our memory abilities, but others argue that it's simply allowing us to focus our limited memory resources on what's most important. Recent studies have shown that people who frequently use search engines show different patterns of brain activation when trying to recall information. Their brains seem to be adapting to the availability of external information sources, developing new strategies for managing the boundary between internal and external memory. This adaptation isn't entirely new. Humans have always used external memory aids, from cave paintings to written language to libraries, but the speed and pervasiveness of digital technology is accelerating these changes in ways we're only beginning to understand. The implications of this digital transformation extend far beyond individual memory abilities. The way we store and access information as a society is fundamentally changing. Traditional institutions like libraries and schools are being forced to reconsider their roles in a world where factual information is instantly accessible. The skills we need to succeed are shifting from memorization to evaluation, from storage to synthesis, from recall to retrieval. But this digital augmentation of memory also raises important questions about human cognition and identity. If our smartphones and computers become seamless extensions of our memory systems, where does the self end and the technology begin? How do we maintain our cognitive abilities while becoming increasingly dependent on external aids? And what happens when these technological systems fail or are unavailable? Neuroimaging. Watching memory in action. One of the most revolutionary developments in memory research has been the advent of neuroimaging techniques that allow scientists to observe the living brain in action. Functional magnetic resonance imaging, fMRI, positron emission tomography, PET, and electroencephalography, EEG, have opened windows into the neural processes underlying memory formation, storage, and retrieval. These techniques have revealed memory networks that are far more complex and distributed than early researchers ever imagined. When you form a memory, activity doesn't just increase in memory-related brain regions, it increases in a coordinated pattern across multiple networks spanning the entire brain. Advanced neuroimaging has also revealed the real-time dynamics of memory processes. Researchers can now track how memories are encoded moment by moment, how they're consolidated during sleep, and how they're reconstructed during retrieval. This temporal precision has revealed that memory processes unfold over multiple timescales, from milliseconds to years. One particularly exciting development has been the use of machine learning techniques to decode memory content from brain activity patterns. Researchers can now use fMRI data to predict what a person is remembering, sometimes with startling accuracy. In some cases, they can even reconstruct visual images that people are imagining or remembering based solely on their brain activity. These decoding techniques have revealed that different memories have distinct neural signatures. The pattern of brain activity when you remember your childhood home is different from the pattern when you remember your first kiss, which is different from the pattern when you remember yesterday's lunch. These unique neural fingerprints suggest that memories are indeed distributed across the brain in systematic, predictable ways. But neuroimaging has also revealed how similar the neural processes of memory formation and retrieval actually are. When you remember an experience, many of the same brain regions become active that were active during the original experience. This provides neural evidence for the reconstructive nature of memory. Remembering isn't just retrieving a stored file, but actively recreating the experience using many of the same neural mechanisms. Optogenetics, controlling memory with light. One of the most revolutionary developments in modern neuroscience has been the development of optogenetics, a technique that allows researchers to control specific neurons using light. By genetically modifying neurons to produce light-sensitive proteins, scientists can turn individual brain cells on or off with the flick of a laser switch. This technology has led to discoveries that would have seemed like science fiction just a few decades ago. Researchers can now isolate the specific neurons that store a particular memory, then reactivate those neurons to artificially trigger the recall of that memory. They can even implant false memories by artificially activating neurons while an animal is in a different context. In one stunning experiment, researchers trained mice to fear a particular cage where they received mild electric shocks. Later, they placed the mice in a completely different, safe environment and used optogenetics to reactivate the neurons that stored the memory of the fear conditioning. The mice immediately began showing fear responses, even though they were in a safe location. The researchers had essentially used light to make the mice remember being in the dangerous cage while they were actually somewhere else entirely. Even more remarkably, researchers have been able to turn negative memories into positive ones and vice versa. By manipulating the connections between memory-storing neurons and reward or punishment centers in the brain, they can change the emotional valence of a memory without changing its content. These experiments are providing unprecedented insights into the neural basis of memory, but they're also raising profound ethical questions about the nature of identity and the possibility of memory manipulation in humans. If memories can be artificially created, modified, or erased, what does this mean for concepts like personal responsibility, free will, and authentic experience? The potential applications of optogenetics to human memory disorders are tantalizing but still largely theoretical. The technique requires genetic modification of neurons, which is not currently feasible in humans for non-medical applications. However, researchers are exploring ways to achieve similar effects using less invasive approaches. The molecular fountain of youth. Another exciting frontier in memory research involves understanding how memories are maintained over time. Recent discoveries have revealed that long-term memories require ongoing molecular maintenance. They're not simply stored once and then passively preserved. This maintenance involves a delicate balance between the proteins that strengthen synaptic connections and the enzymes that break them down. As we age, this balance can shift, leading to the weakening of synaptic connections and the loss of memories. But researchers have discovered that this process isn't inevitable. By manipulating the molecular mechanisms that maintain synaptic strength, they've been able to preserve and even restore memories in aging animals. In some cases, they've been able to reverse age-related memory decline, essentially restoring young-like memory function to old brains. These discoveries are leading to the development of new therapeutic approaches for age-related memory disorders and neurodegenerative diseases like Alzheimer's. While we're still years away from practical applications in humans, the basic research is providing hope that memory loss might not be an inevitable consequence of aging. One particularly promising approach involves targeting the molecular switches that control synaptic plasticity. By developing drugs that can selectively enhance these switches, researchers hope to be able to boost memory function without affecting other aspects of brain function. Another approach focuses on the cellular quality control mechanisms that maintain healthy neurons. As cells age, they accumulate damaged proteins and other cellular debris that can interfere with normal function. By enhancing the brain's ability to clear this cellular waste, researchers hope to maintain healthy memory function throughout the lifespan. Memory enhancement and cognitive augmentation, the same research that's revealing how memories are formed and maintained, is also pointing toward possibilities for enhancing memory beyond normal human limits. Scientists are exploring various approaches to memory enhancement, from pharmacological interventions that boost the activity of memory-related proteins to brain stimulation techniques that can temporarily improve memory performance. Some of these approaches are already showing promise in human studies. Transcranial stimulation, applying weak electrical currents to specific brain regions, can temporarily enhance memory formation and retrieval. Certain drugs that affect neurotransmitter systems can improve memory performance in both healthy individuals and those with memory impairments. But perhaps the most intriguing possibility is the development of brain-computer interfaces that could directly augment human memory. Researchers are developing systems that can record neural activity patterns associated with memory formation, then play them back to strengthen or restore memories. Theodore Berger and his colleagues at USC have developed what they call a memory prosthesis, a device that can record the patterns of electrical activity in the hippocampus during learning, then play back those patterns to enhance memory formation. In animal studies, this device has been shown to improve memory performance by up to 30%. The memory prosthesis works by recording from multiple electrodes implanted in the hippocampus, while animals learn new tasks. The The device analyzes the patterns of neural activity that occur during successful learning trials, then identifies the optimal patterns for memory formation. Later, when the animal is trying to form new memories, the device can stimulate the hippocampus with these optimal patterns, effectively enhancing the brain's natural memory processes. Early human trials of this technology have shown promising results. Patients with epilepsy who had electrodes implanted for medical reasons showed improved memory performance when the device was activated. While the technology is still experimental and requires invasive brain surgery, it represents a proof of concept for direct neural enhancement of memory. While these technologies are still in their infancy, they raise fascinating questions about the future of human memory. If we can artificially enhance our ability to remember, should we? What would it mean to have perfect recall of every moment of our lives? How would enhanced memory change human culture and society? The genetics of memory. Modern genetic techniques are also revealing how individual differences in memory ability are influenced by our DNA. Researchers have identified numerous genes that affect memory formation, consolidation, and retrieval. Some people carry genetic variants that give them naturally superior memory abilities, while others carry variants that make them more susceptible to memory disorders. The COMT gene, which codes for an enzyme that breaks down dopamine, provides a striking example of how genetics can influence memory. People with different variants of this gene have different levels of dopamine in their prefrontal cortex, which affects their working memory abilities. Those with the MET variant have higher dopamine levels and better working memory, while those with the VAL variant have lower dopamine levels and somewhat poorer working memory performance. The APOE gene, best known for its association with Alzheimer's disease risk, also affects memory function in healthy individuals. People who carry the APOE4 variant show subtle differences in memory performance and brain structure, even before any signs of cognitive decline appear. But genetic influences on memory are far more complex than these single gene effects suggest. Memory ability is influenced by complex interactions between hundreds of genetic variants, each contributing a small effect. Moreover, the influence of genes on memory is heavily modulated by environmental factors. The same genetic variant might enhance memory in one environment but impair it in another. This gene-environment interaction means that understanding memory will require not just identifying relevant genes, but also understanding how those genes interact with experience, culture, and other environmental factors. Recent research has also revealed that genetic influences on memory change across the lifespan. Some genetic variants have large effects in childhood, but smaller effects in adulthood. Others have minimal effects early in life, but become more important with aging. This dynamic nature of genetic influences highlights the importance of studying memory development across the entire lifespan. Understanding the genetics of memory is opening up new possibilities for personalized medicine. In the future, we might be able to identify individuals who are at risk for memory disorders before symptoms appear, allowing for early intervention. We might also be able to develop treatments that are tailored to individual genetic profiles. The social neuroscience of memory. Finally, modern neuroscience is beginning to understand the social dimensions of memory at the neural level. Using brain imaging techniques, researchers can now observe how social factors influence memory processes in real time. They've discovered that memories formed in social contexts are processed differently than memories formed in isolation. The presence of other people activates specific brain networks that influence how memories are encoded, consolidated, and retrieved. These social memory networks help explain why we remember shared experiences differently than solitary ones, and why our memories are so easily influenced by the recollections of others. The social brain networks involved in memory include regions like the medial prefrontal cortex, temporal parietal junction, and superior temporal sulcus, areas that are also involved in understanding other people's thoughts and intentions. This overlap suggests that memory and social cognition are intimately connected at the neural level. Research on social memory is also revealing how cultural differences in memory are reflected in the brain. People from different cultures show different patterns of neural activity when remembering the same events, reflecting cultural differences in what aspects of experience are considered important and worth remembering. For example, people from individualistic cultures, like the United States, show greater activation in brain regions associated with self-referential processing when remembering personal experiences. People from collectivistic cultures, like East Asia, show greater activation in regions associated with social and relational processing. These neural differences mirror behavioral differences in what people from different cultures tend to remember about personal experiences. This work is helping to bridge the gap between individual and collective memory, showing how the social and cultural contexts we live in literally shape the neural processes that create and maintain our personal memories. The construction of self-memory is also central to the construction of personal identity. The sense of being a continuous self, the same person over time despite constant physical and mental changes, depends heavily on memory. Your autobiographical memories provide the narrative thread that connects your past, present and future selves. But this narrative construction of self is not passive or automatic. You actively choose which memories to emphasize, how to interpret them, and how to weave them into a coherent life story. Different people can have very different life narratives even when they've had similar experiences, because they've made different choices about how to remember and interpret those experiences. This active construction of narrative identity means that you have some agency over who you are. While you can't change what happened to you in the past, you can change how you remember and interpret those experiences. You can choose to emphasize certain aspects of your story while de-emphasizing others. You can find new meanings in old experiences. This doesn't mean that memory is infinitely malleable, or that you can simply choose to remember things differently. The basic facts of your experience provide constraints on how you can interpret your past. But within those constraints, there is significant room for creative reinterpretation and narrative reconstruction. Understanding the role of memory and identity construction has important implications for therapy and personal growth. Many therapeutic approaches focus on helping people reinterpret their past experiences in more adaptive ways, not by changing the facts of what happened, but by changing how those facts are understood and integrated into personal narrative. The mystery remains. Despite all of our scientific advances, memory remains one of the deepest mysteries of human existence. We can map its neural circuits, identify its molecular mechanisms, and even manipulate it with light and electricity. But we still don't fully understand how the physical activity of neurons gives rise to the subjective experience of remembering. How does the firing of billions of nerve cells create the vivid sense of reliving a childhood summer, the bittersweet pang of nostalgia, the sudden flash of recognition when you see an old friend? How does the reorganization of synaptic connections translate into the rich, meaningful narratives that make up your sense of self? These questions touch on some of the deepest problems in science and philosophy: the relationship between mind and brain, the nature of consciousness, the mystery of subjective experience. Memory sits at the intersection of all these profound puzzles, reminding us that despite all our knowledge, the human mind remains wonderfully, beautifully mysterious. The hard problem of consciousness, how physical processes give rise to subjective experience, is particularly acute when it comes to memory. We can understand in great detail how memories are encoded, stored, and retrieved at the neural level, but we still don't understand how these neural processes create the qualitative, subjective experience of remembering. When you remember your childhood home, you don't just access factual information, you experience a rich, multisensory recreation of being in that place. You can feel the texture of the carpet under your feet, smell the familiar sense of home, hear the sounds of family life. This qualitative richness of memory experience is not captured by our current scientific understanding of neural mechanisms. This doesn't mean that science can't eventually solve these mysteries, but it does mean that we should remain humble about the limits of our current knowledge and open to new ways of understanding the relationship between brain and mind. The ethics of memory. As our ability to understand and manipulate memory advances, we're confronted with profound ethical questions about how these capabilities should be used. If we can enhance memory, should we? If we can erase traumatic memories, should we? If we can implant false memories, how do we prevent this from being misused? These questions become more urgent as memory technologies move from the laboratory to the clinic. Brain stimulation devices that can enhance memory are already being tested in humans. Drugs that can affect memory formation and retrieval are being developed for therapeutic applications. Optogenetic approaches to memory manipulation, while not yet applicable to humans, demonstrate the theoretical possibility of precise memory control. The potential benefits of these technologies are enormous. They could help treat debilitating memory disorders, reduce the suffering caused by traumatic memories, and enhance human cognitive capabilities, but they also raise concerns about authenticity, autonomy, and human nature. If our memories can be technologically enhanced or modified, what does it mean to be human? The ethics of memory. As our ability to understand and manipulate memory advances, we're confronted with profound ethical questions about how these capabilities should be used. If we can enhance memory, should we? If we can erase traumatic memories, should we? If we can implant false memories, how do we prevent this from being misused? These questions become more urgent as memory technologies move from the laboratory to the clinic. Brain stimulation devices that can enhance memory are already being tested in humans. Drugs that can affect memory formation and retrieval are being developed for therapeutic applications. Optogenetic approaches to memory manipulation, while not yet applicable to humans, demonstrate the theoretical possibility of precise memory control. The potential benefits of these technologies are enormous. They could help treat debilitating memory disorders, reduce the suffering caused by traumatic memories, and enhance human cognitive capabilities. But they also raise concerns about authenticity, autonomy, and human nature. If our memories can be technologically enhanced or modified, what does it mean to be human? How do we maintain personal autonomy and authentic experience in a world where memories can be manipulated? How do we prevent these technologies from being used to exploit or control people? These ethical questions don't have easy answers, but they need to be carefully considered as memory technologies continue to develop. We need frameworks for thinking about the appropriate use of memory enhancement and modification technologies, guidelines for research and clinical applications, and protections against misuse. The future of human memory. As we look toward the future, it's clear that human memory will continue to evolve, both through natural processes and through technological intervention. The digital age is already changing how we store and access information, and these changes are likely to accelerate. Brain-computer interfaces may eventually allow direct integration between biological and artificial memory systems. Genetic therapies might allow us to enhance memory abilities or reduce susceptibility to memory disorders. Pharmacological interventions could become routine tools for optimizing memory function. But perhaps the most important evolution in human memory will be cultural rather than technological. As we better understand how memory works, we can develop better strategies for learning, teaching, and preserving knowledge. We can design educational systems that work with rather than against natural memory processes. We can create cultural practices that support healthy memory function across the lifespan. We can also become more sophisticated consumers and creators of information, understanding how our memories are shaped by the stories we tell and the narratives we encounter. We can be more thoughtful about which memories we choose to preserve and pass on to future generations. A gentle conclusion. And so, dear listener, as you prepare to drift off to sleep tonight, remember that your brain will continue its quiet work of memory formation and consolidation. The experiences of today will be woven into the ongoing narrative of your life, connected to memories from years past and integrated with hopes for the future. Your memory is writing the story of who you are, one neural connection at a time, one synaptic change at a time, one dream at a time. It's a story that's uniquely yours, but also connected to all the other stories being written by all the other minds that share this world with you. The memories you form tonight, the dreams you'll have, the thoughts that will drift through your consciousness as you fall asleep, all of these are part of the ongoing creation of yourself. Your brain will sort through the day's experiences, determining what's worth preserving and what can be safely forgotten. It will form new connections, strengthen important pathways, and perhaps even generate new insights and understandings. This process isn't just about storing information, it's about creating meaning. Your memory system doesn't just preserve facts about what happened, it creates narratives about what those facts mean. It connects your experiences to your values, your goals, and your sense of who you are and who you want to become. Sleep well, knowing that your memory system is working tirelessly to help you make sense of your experience, to learn from your past, and to prepare for your future. And when you wake tomorrow, you'll carry with you not just the memories of today, but a slightly revised, slightly updated version of the beautiful, imperfect, wonderfully human story of who you are. Remember that memory is not just about the past, it's about the continuous creation of self in the present moment. Every memory you form, every story you tell about your experiences, every meaning you find in your past. These are all acts of creation, ways of actively constructing the narrative of your life. And in this construction, you are not passive. You're an active participant in the creation of your own story. You get to choose what memories to emphasize, how to interpret your experiences, and what meanings to find in your past. This is both a tremendous responsibility and a tremendous gift. Sweet dreams, fellow travelers in this strange and wonderful journey of consciousness. May your memories be kind, may your dreams be meaningful, and may you wake refreshed and ready to write new chapters in the ongoing story of your life. Remember that you are not just the product of your memories. You're also their author, their editor, and their interpreter. In the quiet moments before sleep takes you, perhaps you can appreciate the remarkable gift of memory, this ability to transcend the immediate present, to connect with your past self, to imagine your future self, and to find meaning in the continuous flow of experience that makes up a human life. Your memory is one of the most remarkable achievements of evolution, a system so sophisticated that we're only beginning to understand its true complexity and beauty. Treasure it, nurture it, and use it wisely. For in the end, we're all made of memories, and in our memories, we live forever.